So welcome again, everybody. It's nice to see lots of people here. My name is Matthew. So I'm the team lead for the BioImage Archive at EMBL-EBI, and I'm going to tell you about the archive today. Many thanks to AJ for the introduction. In terms of what you should be expecting to get out of this session, by the end, you should have an understanding of what the types of imaging data you can deposit into the BioImage Archive and how you'd go about doing that. You should be able to understand what types of data we have and how you can access those data. And finally, and in some ways, perhaps most importantly, where you go to find out information. And we'll be covering this at a reasonably high level. It won't be a detailed walkthrough of the submission process, but it should be enough to know if perhaps you're expecting to submit data in the future, to be aware of what will be coming up. To go into just a little bit more detail on that outline, I'll start by giving some background on the archive itself, particularly why it was created, what the overall purpose of the project is, and how it relates to some of the other community imaging resources that exist. Then we'll cover browsing and accessing data first before we look at the submission process. And the reason to do it that way round is that it's easier to understand how the submission process works, and particularly how the information that you enter during the submission process will be used if you've seen how that will be presented when submission's complete. And the last sections of the talk will be quite short. I'll give a very brief overview of what we're working on at the moment, what the upcoming developments are, show you where to go to look for more information, and then we'll have opportunity for questions at the end. So the BioImage Archive is one of the newest archival data resources at EBI. And the overall purpose for the BioImage Archive is to take bioimaging data from any modality, and bioimaging is a very, very broad field with lots of subdisciplines. So this is lots and lots of different types of imaging. And there are four main reasons to do this, four sort of primary goals behind the BioImage Archive. The first one is to improve scientific reproducibility of results that rely on imaging data. So imaging has been used in science for a very long time, but increasingly that dependence is quantitative. And where scientific results depend very directly on the results of, for example, analyzing images, we can't really trust those results unless we can go back to the original images that underlie them to check that the process by which those results were arrived at made sense. And if you want to be able to reproduce those results, if you want perhaps to be able to carry out experiments again, it's also important to have access to the original data. So that's one of the reasons. Another reason is to provide a platform for hosting reference data sets. And reference data sets actually will come up a few times during the talk. So reference data sets are data sets which are likely to be a very wide value to a large cross-section of the scientific community. For example, projects such as cell atlases, where there's a long program of work, there's lots of sort of time and effort, and therefore effectively money goes into producing large complete data sets. And because of that, it's important that we can make those data sets available as widely as possible for as long as possible. And then two sort of closely related reasons to have an image archive, both related to reuse of data. So one way to reuse data is to be able to get new scientific results out of existing data sets. And that is important. Sometimes this will be being able to do perhaps some preliminary research by looking at images and thinking if there's ways in which they could be used differently from the purpose for which they were originally intended. But another sort of reuse direction is that modern sort of biological image analysis is very dependent on having reference data. Sometimes that's just baseline benchmark data, but particularly deep learning AI techniques really need large corpuses of well annotated reference data, particularly if you want to, for example, train new models. And having a platform that can host those is therefore pretty important for sort of helping to drive the development of image analysis techniques. There's another reason to have a primary image archive, and this relates to a concept that I think originally arose in the genomic world, whereby we can sort of roughly divide biological data resources into two different types. And one of those types is primary archives. And primary archives are really there to take data, hold that data as faithfully as possible, so to keep it as close to its original raw form as possible, sort of index it, take the right types of metadata that will be needed to make that data understandable in the future, and then present that out to the community. But on top of that sort of base level platform can sit what we sometimes call added value databases. And added value databases can do more with data, perhaps through expert curation, because they have sort of strongly scientifically trained people with the right background, perhaps by integration with different types of genomic data, so from different imaging modalities or imaging in genomic data. And these two resources need to work, or types of resources need to work together. And I'll talk about two resources which are a little bit like added value databases. And the reason I say a little is partly because of the history, which I'll get into in a minute. And those are the IDR and EMPIAR. So the first of those, the image data resource, is an added value database that was developed by the OME. So OME stands for Open Microscopy Environment, who are a group that do a lot of work in image data management. And they developed the IDR originally in collaboration with EMBL-EBI. And the IDR is hosted at EBI in the Embassy Cloud, but is run by the OME team. And the IDR is there specifically to take reference data sets. So these data sets I mentioned that are likely to have wide scientific value. And the IDR can provide their sort of expert curation expertise and presentations of those data in order to really enrich them as far as possible. Another related resource is EMPIAR. So EMPIAR stands for the Electron Microscopy Public Image Archive, and EMPIAR is another EBI-based resource. And EMPIAR originally arose to hold raw images that underpin 3D cryo-electron microscopy maps, because again, from the reproducibility aspect, to go back from a structure derived from cryo-EM through to the electron density map, through to the cryo-EM raw images that produce that density map, requires access to those raw images, and there was no resource for that. So EMPIAR was created originally to fill that gap based on community need. EMPIAR has, I think, because it's been so successful, now expanded to take other types of 3D electron microscopy data, particularly volume EM. And there are various types of volume EM data, which I'll mention briefly later on, because exactly where data should go depends a little bit on your type of data, and EMPIAR is one of the reasons for that. So just to put the resources into historical context, EMPIAR is the oldest. It launched in 2014 and is now 7 years old. The IDR launched in 2016, 2017. The BioImage Archive launched 2 years ago in 2019, but this is the first year for which we've had a dedicated development team to run it, which is part of the reason why this webinar was happening now. And so the BioImage Archive is the youngest of the resources, but it's actually growing quite rapidly. And I should mention one more data resource, because it will come up as I go through the rest of the talk, and that's biostudies. So this is another EBI resource, and biostudies is a very, very general data resource designed to hold data that can that don't necessarily fit into one of the other structured archives at EBI. So for example, raw sequence data would go into the ENA, the European Nucleotide Archive, but something that wouldn't fit into the ENA could perhaps go into biostudies. And biostudies is designed to take those different types of data and link them together. So it has a very general mandate. And the biostudies was the platform in which the BioImage Archive originally was developed and is still being developed on top of biostudies. So in a sense, the BioImage Archive is a layer on top of biostudies. And the reason to mention this is that sometimes as you go through aspects of the submission process, you'll see biostudies directly because it's in a sense the back end. So now I'm going to look at how you can access the data in the archive. And to do this, rather than go through slides, I will show you the website for the BioImage Archive itself and show some examples of how you can look for data. So from the website, you can browse all of the data sets we have in our collections. And this is the interface for browsing data and also showing search results, which I'll go into in a moment. And on the right-hand side, we're seeing the names of different data sets together with a little bit of information about those data sets, including things like their release date. And actually, this is sorted by release date. So the most recently released data sets are at the top of this list. You can change this sort order. And this is showing everything that we hold or index. So a little over 1,200 data sets. You can filter the information that you're seeing by different things. You can filter by year. You can filter by, for example, file type. One of the things that is potentially useful to filter by is this collection category. And this collection category comes out of the fact that the BioImage Archive has really 3 different types of image data. There are data that were imported from other data resources. So for example, this JCB, this stands for Journal of Cell Biology, and that's a collection of data that came from something called the Journal of Cell Biology Data Viewer, which was a data hosting platform associated with the Journal of Cell Biology publication. And that was decommissioned a few years ago, and then the data was sort of taken on by the BioImage Archive. And one of the roles of the archive is to be able to do that, to be able to take data which perhaps originally had a different hosting platform, but that wasn't sustainable in the long term. The next collection, which is actually labeled BioStudies Other, so that's where this BioStudies links come in, is data that's submitted directly to the BioImage Archive. And the final collection, EMPIAR, are data sets where the actual data sit in EMPIAR, the resource I mentioned a little bit back, but we index the data sets so that you can find them by search, so that you're searching across all of the image data that we know about rather than just a subset. So I'll show how that search process works next. So there's a few different ways you can search. If you're looking for something very specific, so for example, if I know the name of a data set or an accession and all I want to do is find it, we can search just by that name. And we'll see, in this case, there's one result, and we see both the name of the data set and a little bit of information about it. We can also search by keyword. So suppose we're interested perhaps in browsing rather than looking for something specific, and I search for the word brain. So then we see more different results from search, and the 2 things to note about the search. The places where the search term that we search for come up are highlighted in yellow, but you'll also notice the sort of orange colored results. And that's because the search system will look for results which are related, or will search for terms specifically that are related to what you search for. So in this case, we're seeing results for brain, but we're also seeing results for hippocampus, which is a subcomponent of the brain. And that's useful because, for example, this result towards the bottom of the results mentions hippocampus, but doesn't mention brain. So sometimes you can find things that are related to your search term, and that can be useful. And again, you can filter the results by year or collection. So if you were looking only for example, results in EMPIAR, you could filter just to see the EMPIAR results. You can combine search terms to make more advanced searches. So suppose we wanted just things that mention both brain and Drosophila. So assuming I can spell correctly, we can do a search and find accessions where Drosophila and brain are both mentioned in the metadata associated with the accession, which is what we're seeing. And again, actually, we see some related terms related to both of the search terms. And we can construct more advanced searches. So if we wanted to search for accessions which had the word brain but didn't specifically mention Drosophila, so in a sense we wanted to rule out some of the searches, we could do that. And you can actually build up quite complicated search terms using regular expressions and multi-part compound queries, which I won't go into now, but there is more information about on our help pages, which I will touch on towards the end. So the search system is quite flexible. Let's look at a specific data set. So far we've been seeing summary information about multiple data sets. If in one of these searches we click on a name, we'll see the information for that data set particular. And this is really split up into two parts. The part on the left-hand side, which is the general metadata to do with the accession, and the part on the right, which is to do with the individual image files in that accession. So on the left-hand side, we see the metadata and the information that's associated with the whole data set. So we see the title, there is a description, which is usually an explanation of the experiment that gave rise to the data set. Then we see more information, in this case, things like the organism that was being studied, or organisms in this case, and then some more information. And actually, what goes into this is dependent on the level of detail in the submission or that the submitter provides. And that's why it's useful to explain this before we touch on or go through how submission works. In some cases, information is indented and hidden, and you can expand that. So the protocols for cell cultures, for example, are actually here, although they're not immediately visible. The image data side is a little bit different. So over here, we're seeing just a small subset. We're seeing 5 images from the data set that we're looking at. But we can already see down here that there are actually 127,000 images in the data set. So it's useful to have a way to browse and filter that result a little bit further. And we can do that by expanding this panel here. So if I click on the button, little square that I just clicked on, we see metadata that's associated with each individual image in the data set. So these are things that differ from image to image. And in this case, this is a high content screen. So the relevant information for each image is to do with the compound with which that particular sample was treated. And this metadata differs from accession to accession because different types of imaging experiment really have different requirements. If this was a genetic knockout screen, it would make sense to describe, for example, the genes that were being knocked out. If this was a cross comparison looking at multiple organisms, this would explain what the different organisms were. And there is quite a lot of information here. In this case, we have things like Unichem links, again, because it's relative to the compound. We have formulae, and this information can also be searched. So this plus icon here opens up search boxes for each of these categories. And I can, for example, then search for caffeine in the compound name, and I can then find all of the images which were treated with caffeine. And this is useful when it comes to finding particular images, particularly in large data sets like this one, where there were several thousand images, but then also in getting hold of downloading those images. And I'll talk about download next. So we have a few different ways in which you can actually access the data. There is a button here, download all files, and that will give you every single file in the data set. That can be a very large number. So in this case, there are nearly 127,000 images. I think our biggest data sets have a few million. For that reason, it's useful to be able to download subsets of the data set. And you can do that in a couple of ways, one of which is you can select individual images and download them. That is viable for smaller data sets. It's less convenient for very large ones like this. You can download single images simply by clicking on them, and that can be useful just if you want a preview of one of the files in the data set, if you'll want to check whether it's the type of data that you actually want, for example. But you can also couple the specific metadata search that I showed you with the download system. And so at the moment, the interface is showing me 9 files out of 127,000. And if I select just those files, I can download to just the 9 files, in this case, where the relevant compound was caffeine. So you can combine the image level metadata search with the filtering on these things, which are called facets, to get at the information that you particularly want. If you need to access data at a larger scale, so if you do need to download either whole data sets, particularly for large data sets, where individual file download, particularly by HTTP protocol, might be slow, or if you need multiple data sets, we provide a couple of options there. One of which is that each data set has an FTP link, and what happens when you actually click on the link will depend a little bit on your web browser, because different web browsers handle FTP links in different ways, but you can supply that link to an FTP client and use FTP file transfer protocol to either download whole data sets or subsets, because you can navigate the raw data files. We also provide data access by something called Aspera, which is a much faster data access protocol, but does require a little bit more setup time. So if you do want to download multiple large imaging accessions, it's worth the time investment to set up Aspera, but that will take a little while because it has specific network requirements. So that's a short overview of how you look at, browse, and access data. Now I'm going to move on to the submission side, what happens when you want to put data into the archive. Before I go into any detail, briefly on the sort of motivational side, why should you submit your image data to an archive? There are lots of reasons. I picked 4 particularly, which are all slightly different. Perhaps the most compelling one is that it's the right thing to do scientifically. So when I mentioned the founding goals of the BioImage Archive, scientific reproducibility is sort of one of the key pillars of science itself. And we need open access to the data that underlie scientific results really to make science work. So providing your imaging data, in fact, providing whatever data accompanies your publications, if you're publishing results, it's the right thing to do. There are other reasons, though, one of which is that it's often, it requires a lot of time, resources, and skill, really, to produce imaging data. And publishing the data, even independently of the scientific results, highlights that. It gives publicity to your data, independent perhaps of the manuscript, and that can be quite valuable, particularly to demonstrate when there are new techniques, and what you actually want to do is give people access to the data as soon as possible. Another reason, and if you've ever had to manage data within an institution in the long run, that this one might appeal to you. This takes away the need to worry about long-term preservation of your data, or at least some of your data. So as I'll get onto in a moment, the BioImage Archive is really for data associated with publications and reference data. So it's not there to provide a data hosting platform for sort of everything that comes off imaging instruments in your institution. It's not there for pre-publication data management. We can't solve that side of the problem, unfortunately, just because of the scale that would be involved. But we can worry about what happens for long-term preservation of image data, and that can actually be quite challenging because often you will, particularly in the research world, move between institutions. Often, when you've done the experiment, you'll have immediate access to the data, but in 5 years' time, you might lose that access. And having that in a safe place that we can provide is quite a substantial long-term advantage. And final reason, and this is perhaps more of a push reason than a pull reason, that increasingly journals and actually also funders will wish, and perhaps more strongly than wish, they will mandate that you submit your image data and you publish your image data alongside your manuscript. So that, as I said, it comes at both the publication side, but now, depending on funding route and funding authority, you'll often have to write a data management plan or something that will explain how you will make your data accessible to the scientific community, which really comes from the first reason that this is the right thing to do. And it's something that's good for funders because it maximizes the value of expensive research data. So, okay, you have been convinced by one of these arguments. You want to submit some image data. How does the process work? I'll go through the steps, both at high level and then covering each one. This won't be a detailed walkthrough, but it'll give you enough information to know what's coming up if you intend to do this at some point. It may look a little bit daunting at first, but really the fundamentals of this are checking that you're submitting the data to the right place, transferring over data, and then providing metadata. That's really all there is to it. So the first stage of that, checking that you're submitting data to the right place, the BioImage Archive takes data from any imaging modality. We're not specific to light microscopy or light and electron microscopy. We will take anything that's bioimaging in any format. So if you have worked in bioimaging, you will know that there are a very, very large number of different data formats, and actually we can accept any of them. Our main constraint, as I mentioned earlier, is that data should be associated with the publication. So one of the primary use cases for the BioImage Archive is that you're looking for somewhere to host your data when you're at the publication stage of the sort of life cycle of your experiment. But we will also take reference data sets. So if you think you have a reference data set that isn't necessarily immediately associated with a publication, please do get in contact with us to talk about that. But also think about the IDR, the image data resource, because they have a very good sort of methods for handling reference data sets. And finally, the main thing to be a little bit careful of in scope is that certain types of electron microscopy, particularly 3D electron microscopy, should be deposited in EMPIAR, because actually, again, EMPIAR can do more with those data. And there are more details to help you work through whether you're doing what the right place for your data are on our scope page. So this in particular, when you navigate to this page, it will tell you which types of data should go into EMPIAR. And we're probably going to expand on this a little bit and build it into the submission system just to make this even easier. So now you're at the stage where you know that you're submitting your data to the right place. What's the next stage? The next stage is to think about exactly what you're going to submit. So usually when you do an imaging experiment, you'll have some raw data files, but you might also have other things. If you've done analysis of your data, you will often have the results of those analyses. You might have intermediate data. You might have supporting files. So if you're doing things in 3 dimensions, you might have 3D meshes and so on. Any of that can go into the BioImage Archive. And in fact, we're keen that the data sets are as complete as possible. And by complete, I mean that somebody else should have all the information and pieces that they need in order to understand your data. So think a little bit about what that means in terms of should you include some of the intermediate stages of your analysis if somebody else will find it very difficult to reproduce that analysis without it, and include those. And then organize them locally into some kind of sensible sort of directory and file structure at your end. Then the next stage is to register an account with us. So this is Very, very simple. All we really need from you is a name, an email address, and a password. You can also supply an ORCID ID, which is a good thing to do to help track use of your data. The reason why you have to register early on in the process is because when you do that, personal file space will be created for you at the EBI, which allows you to upload your data before finishing off the submission. So this step comes early because it then lets you go on the next step, which is getting the data over to us. The methods by which you can get data over are actually very similar to the methods that you can download data. So the submission tool, we have an online web-based submission application. You can upload data through that tool. And if your data set or the individual files in your data set aren't too large, where too large means more than about 16 gigabytes per individual file, that will be easiest because it's an intuitive sort of point and click process that you're guided through. If your data set is larger, so if your data set is sort of into the perhaps hundreds of gigabytes size, or you have very large individual files, you'll need to use one of the other data transfer mechanisms. And the two that we support at the moment are FTP again, File Transfer Protocol, and Aspera. And similarly to downloading data, these are roughly ordered in how difficult they are to use. So FTP is a little bit more involved than using the submission tool. Aspera is harder, it has more setup time, but it is much faster if you can get it set up. Also importantly, and this can be very important, particularly if the reason that you're making a submission is that you're trying to publish a manuscript very, very soon, and perhaps your publication wants you to have an accession identifier for image data associated with that. It will take some time to transfer very large amounts of data, depending on largely how fast the network connection is at your end. So if you have hundreds of gigabytes to multiple terabytes of data, you will need to allow potentially multiple days in order to transfer over data. So sometimes this can be done at sort of with 24 hours notice is about the fastest you can do this process, but it will take longer if your data set is very large. Then the key component of submitting data to the BioImage Archive, which is a little bit different from some of the other archival systems, for example, at the EBI, is that you create something called a file list. And a file list, which is actually just an Excel spreadsheet or one of a few other file formats, is something which tells the submission system what files you want to include. So I mentioned that you'll have a personal file area at the EBI that you can transfer data to. The file list tells the submission system which of the files in your personal file area are in your submission. So it's a little bit like an index or a manifest. It also lets you specify what the image level metadata associated with your images should be. And that's where the information that I showed earlier, for example, in the high content screening data set with different compounds per image, that's where that comes from. So to illustrate this, the top left part shows how your files might look on disk. So I think the bottom is an example from Windows and the top is an example from a Mac. You have 4 image files, for example. Practical data sets are much larger, but this is a small example for reference. You would create an Excel spreadsheet, and the Excel spreadsheet would list the files that you want to include in the submission. And then it can also list attributes and values associated with those files, and that's that file level metadata. And the reason I say attributes and values something more rather than something more specific is because, as I said before, this varies from imaging data set to data set. So some are about what was the relevant gene, some are about what was the relevant organism or tissue or cell type or compound. So there's actually a lot of flexibility to describe the image data in different ways, and we need that flexibility because of the breadth of different types of imaging experiment. And if you're unsure about what to include, it's probably a good idea to contact us because we can make recommendations and show examples of where different sort of subdomains of bioimaging have different standards. And then when you go through submission, this bottom pane is actually a screenshot from the browsing interface. So it's the same browsing interface I showed before. And this shows how these things will appear. So the attributes and values you specify in your Excel file together with the images are what will be shown to whoever is looking at your imaging data when you've finished. So the final part of the submission is giving the rest of the metadata. So it's giving the metadata that is associated with the whole data set. And you do that through a web form in the submission tool that I mentioned earlier. And probably the most important thing to remember about the web form is that when you start the submission, you'll be asked what type of data you have, and the phrasing is a little bit confusing. It's what is the study's parent collection, and this comes out of the fact that, as I said before, the BioImage Archive is based on biostudies, and it's important to select bioimages because that will make sure that the submission system asks the right questions, and it asks those questions by giving you a template to fill in, or a web form to fill in. And actually the first time you register as a new user, you'll be given the option to look through a short video about how to fill this form in. It's fairly intuitive. It's mostly entering text into the different fields. Some of the fields have drop downs for options where there are existing ontologies, so a way to describe sort of information that relates usefully to other information. For example, for different types of imaging, you can actually always override those. So if none of the options for imaging technology type make sense, for example, because you're doing something new, you can specify your own value. And probably the most important thing when going through this, because there's quite a lot of flexibility into how much detail you provide, at least at the moment, think about who is going to be looking at your data and try to give them the information that they will need in order to interpret it. And then final part of the process, once you've filled in the web form, you press the submit button, at which point it'll be validated. So the submission system will check that you've given the right information. And at the point at which you submit, then your data are then copied from your sort of personal file area at the EBI to permanent long-term archival storage that's efficiently backed up and there for the long-term future. This doesn't by itself automatically make your data available to the whole world. When submission really means that you've created a complete package of data and metadata and it's moved to the right type of storage, you choose when the data are released. So you can set a release date, and that's the point at which other people are able to view your submission. And the reason to separate these things out are that you will often want your data to be released, to be visible, say when the publication associated with the data comes out, or at some other particular time. You have control over when the rest of the world gets to see your data, and that allows you to do the submission of the data pre-publication, which, because of those potentially longer timescales for larger data sets I mentioned, is important. You can also share the submission privately before release. So before it's publicly available, you can share it with other people. Main use cases for this tend to be sharing with, for example, reviewers or editors. Very good question, actually, and I'll answer those now rather than wait to the end. In terms of are the reviewers, there is no sort of curation process from the BioImage Archive end. The submission system validates information, but there's no sort of human in the loop curation. And what that means actually is that you can get your data out very, very quickly if you need to. We can provide an accession identifier the next day if you can get the data to us fast enough. Second, very good question, very important question. This is all completely free. There is no charge for using the BioImage Archive, and there won't be either for uploading or accessing data. Final thing I should say on this separation between submission and release, you can extend release dates. So it is sometimes the case that you are preparing for a publication, you get your data uploaded, you finish the submission, you want your data to be released at the same time as publication, and something delays publication, you can then go and change the release date so it's further into the future and lines up again. It is actually also possible to make some updates. So for example, if you've made a mistake during submission, if you need to update and add a publication reference. So one situation for doing that might be you release a preprint and you release the data with a preprint, but you want to go back and add the finished publication. You can do that as well. So some updating of metadata after submission is possible. We can, if necessary, be quite flexible, but we do have to make sure there are good reasons for flexibility in updating existing submissions, because where they're linked to by the outside, we can't have the information change too much. So that's the bulk of the browsing and submission process. I'll finish off briefly by just explaining what we're working on. So the major things that we're doing at the moment, which will be coming over the next few months, are giving a little bit more sort of direct support through the submission process, just to make that easier. We will be working on a new metadata model, and that'll be the next slide, so I won't talk about that immediately. And finally, we're looking at how we sort of integrate both submission guidance and submission systems with other imaging resources, particularly with EMPIAR and IDR, which I mentioned, because sometimes it can be a little bit confusing, particularly if you're doing something like correlative microscopy, where you might want different types of data going to different places. Understanding how that process works is something we'd like to make smoother. Again, I'll take a question now rather than at the end. I should have mentioned this earlier, but yes, there is API access to the data. So we share the underlying biostudies API, and the API and the sort of links to instructions for the API through the help pages. And the API lets you browse data sets, it lets you search, and it also lets you get access to individual files. So actually API development is something, further development of the API is coming, but there is a base level already there. So I mentioned metadata several times. Metadata in bioimaging is quite complicated, and it's complicated again because bioimaging is so diverse, because it covers really lots and lots and lots of different related techniques, which can be very different indeed. So for example, some techniques involve, many techniques involve having a camera to capture photons or potentially to capture electrons, but some don't. So atomic force microscopy, for example. So that means that a general metadata standard that said, please specify what camera you had wouldn't make sense. To try to bring some standardization to this rather complicated situation of lots of different sort of types of data, a number of community working groups came together at a workshop a couple of years ago and came up with a standard called REMBI, which is short for Recommended Metadata for Biological Images. And that the figure from REMBI, and REMBI was actually published earlier this year, again as a set of guidelines rather than a very specific schema, breaks that metadata down into different components. So metadata, for example, associated with the sample. So what is the thing you are imaging, specimen, which is perhaps how you've prepared it, how you capture it, what the data is, and so on. And so quite a lot of what we're working on at the moment and will be working on over the next few month is implementing REMBI as the metadata standard for the BioImage Archive, and that will also actually expand to the other resources. Looking a little bit further ahead, there's lots of really exciting work going on about new data formats for imaging data, particularly for very large images and images where you need to access parts of an image. So another thing that makes bioimaging a little bit more sort of complicated than some other areas of science that generate data is that often you'll want to be able to access, say, one particular channel from a multi-channel image, or you'll want to be able to access one time point from a time series, and the whole time series might be represented as one image. So you need access to sort of subcomponents of the image, and new data formats provide a really good way to do that, particularly at scale. So we're working on how we integrate those into our existing infrastructure. On top of that, we want to build facilities, or perhaps better facilities, for interactive viewing and visualization of images, because actually we can't provide much ability to visualize images before you download and explore them, really because of the diversity of data types. So we need at least behind the scenes to be able to do some conversion to a standardized format to enable that. And then back to APIs, we'd like to be able to provide some of this image specific information, like finding individual channels in an image or time points through an API, but that's actually coupled to the work on image formats. So last slide before we go on to questions, and at that point I'll answer the question that I haven't covered so far, and then you're welcome to ask more. Where to go to look for help? First port of call is our web page. There's a help section there, which I'll show on the next slide, that covers a few sort of commonly things that are particularly require further explanation, like the search system. There is also a more detailed frequently asked questions linked from there. And finally, we have a help desk that's usually fairly responsive to new queries, BioImage Archive at ebi.ac.uk, and we're always very, very happy to talk to you through that route. So this is the web page, which again has the relevant links in it, so you don't have to write down or remember them, and some more information about the things that tend to be particular challenges. Just before we go on to questions, I should take the opportunity to plug a couple of things. So firstly, AJ mentioned at the beginning, nearly at the end of the webinar cycle for this year, but do look at upcoming EBI webinars. But you can also look at previously recorded webinars. And actually, I did a lot of that when preparing for this webinar and learned lots of interesting things myself. So I'd strongly recommend doing that. And final plug, we are running a course in May next year that combines image analysis and sort of modern ways to analyze biological image data, particularly sort of exciting AI things, together with the data resources from which you can get images, like the BioImage Archive and other resources. Registration for that course closes in January, and the course itself will run in May, so please do go and check it out. So let's finish off with questions, and I'll answer the question that I haven't answered that came through the chat, which is for 3D CLEM, so that's correlative light and electron microscopy data sets. Will accession numbers link the projects between EMPIAR and the BioImage Archive? The answer to that is yes. We actually have a specific deposition protocol for CLEM, or in fact general correlative data, and actually the figure I showed that shows the overview of how to deposit data is from that paper. It does mention biostudies rather than BioImage Archive because it predates the BioImage Archive, but that does explain how you get the different parts of the data into the right places, and then how we can link together the accession numbers in the right way. So another question, the dataset search, is it looking into experimental protocol descriptions? Yes, it looks at all of the text in the metadata anywhere. So it will look for anything that's, if there's a phrase inside the experimental protocol description, it will find that. And there was the, just to reiterate, is submitting images free of cost? Yes, submission is completely free, access is completely free. There is no charge for anything unless you're a research funder, in which case we do get infrastructure from somewhere, but there's no cost to end users. And you can submit confocal microscope images. Yes, absolutely. That's one of the most common data types because it's one of the most common data types in biological imaging. We will take any type of biological imaging data, including confocal images. You can think, can I submit the images? So if they're not going to be part of a publication, we need to have a justification for why there's benefit to releasing them to the public, We can be quite flexible about that. So good reasons are if you have a particularly sort of scientifically important data set, if you are trying out a new technique and it's important to make that data available to the community. So the answer to that question is yes, sometimes, but the best way to do that is to get in touch with us to discuss it specifically. And the two other questions I think we have open, I did briefly cover, but again, we do have an existing API that's really comes from the lower level biostudies, but we are building something that's a little bit more image specific. And there isn't, from our end, there isn't a sort of human specific review curation process. There's automated validation of data and metadata, but there's no sort of curation queue that's required in order to unlock the images. So those are all really good questions. Thank you very much. I'm happy to answer more if any come up. Can a submission be updated once made public? Is it versioned? Yes to both of those things. So submissions can be updated. If possible, it's good to get in contact with us if you want to make substantial changes because of other external participants that might have sort of linked to images or linked to data sets. And yes, behind the scenes, all changes are versions, so we can actually revert to old versions. That reversion process is not automatic though, so we would need to get involved. That's something we're looking at, particularly because we're looking at how we might be able to offer DOIs, but we can offer DOIs, but we can't offer DOIs automatically. And one of the reasons for that is because it's a little bit more complicated where you have different versions of the data set. Would there be a preference of next generation file formats, NGFFs, in the future? Yes. So actually the reason I didn't mention NGFF specifically, because if you're not in the sort of bioimaging data world, the term might not make sense, but this is the new way to represent bioimaging data that's more sort of amenable to large scale analysis and large data sets that I was talking about in terms of our future developments. And NGFF provides a lot of good opportunity for us to be able to offer data in more useful ways and for us to be able to offer easier ways to visualize. So yes, I think we will have a preference towards submission in NGFF format, but we want to keep submission formats flexible because I think particularly for people that don't have much experience with converting image data, it's too high a bar to at this stage require that submitters have to do their own image conversion. Also very good questions. Nice to hear about things that I was trying to cover.